Thanks so much. Okay, so I really appreciate the opportunity to speak to you all about this. I am at very short notice replacing Melinda Mills, and I'm disappointed to not be able to hear Melinda Mills, and I can only imagine you will feel the same way. Big shoes to fill. I'll try my best. And I thought that the Sydney Brenner quote was very apt, because we're going to be talking about stuff that's happening very recently, which we have a very sharp understanding of, and trying to put it into the context of the legendary mist, which which I certainly don't have a good grasp of. And these last couple of days have moved the ground beneath my feet a little bit. So what we're going to talk about is the way in which Genome Wide Association studies are being conducted today and where they've come from. So Genome Wide Association study has an objective to identify the positions in the genome that are related to a complex trait or a disease. And that sort of links to a broader objective in medicine when we're looking at humans, which is trying to understand the etiology of complex disease and also to develop predictors of complex disease. And so Genome Wide Association studies aim to be a tool that contributes to those two endeavors. So what I'm going to try to do, and I may fail in doing this, is try to talk about how the laws, the Mendelian laws that we've talked about the last couple of days, map to the exercise of gene mapping as it is done in contemporary research. And I just wanted to start with a bit of a context, which I think I take a lot of pleasure in thinking about, and it might not actually be that interesting to others, but there's this beautiful paper, there's a perspective piece by Yuri Lazetnik written in Cell in 2002, and he was just starting out as a sort of a PI somewhere in America. And he's a cell biologist studying apoptosis, and he writes this sort of semi-facetious question, like, can a biologist fix a radio, right? And he poses the problem the biologists face, that there's some unbelievably complex system that they're trying to understand, cells or entire organisms. And the tools that they have to try to understand them are rather blunt. So the example here is if a biologist used their tools to understand why a radio is broken, well, first what you would do is you'd start with 1,000 completely fine radios, and then you'd either systematically or randomly introduce perturbations into each of those radios. And you'd painstakingly map what the consequences are of each of those perturbations onto the function of the radio. And hopefully you could start to build an understanding of the circuitry and the components and how they all work together. And this isn't something which is to denigrate molecular biologists, it's to celebrate them. It's unbelievably difficult to do this. Now, the reason that I talk about this is because as statistical geneticists, we're at an unbelievable advantage when we compare ourselves to that scenario, because we have front row seats to an amazing natural experiment, which is happening continuously and has been throughout history. So what we have is a human genome, which is about 3 billion base pairs in length. And every new birth has about 70 new mutations. And there are 170 million births per year. So every single year, on average, and ignoring sort of technicalities like some of these mutations might not be viable, etc., about every position in the genome gets mutated like about 3 times. This is an unbelievable factory of experimentation that we can simply observe by sequencing or genotyping individuals. And so this is what happens to those mutations. So this is a plot which basically shows the fate of a mutation when it comes into the population. So it's not really coming to the population at 0, it's at 20% when it starts, but some of them are just going to go to extinction very quickly. And some of them might be lucky enough that they persist in the population and the frequency isn't at 0. And so they need to be transmitted from one generation to the next. And so over time, we've been able to sequence more and more individuals, and we get a sort of a glimpse into like what actually is polymorphic in the population. We have a good understanding of how many variants there are in the population, in different populations. In Africans, there are about 15 million positions in the genome which are common polymorphisms. That means that one copy of the allele is present in at least more than 1% of the population. But as we looked at, if you just keep sequencing people, you'll eventually find every position in the genome is polymorphic. And so the bar on this side is limited by the sample size. The more you sequence, the higher this bar is going to go. We're not going to find more common polymorphisms. So this is, if we believe that genetic factors influence complex traits, this is where you look amongst the polymorphisms, because that is what gives you differences between individuals on a genetic basis. Okay, in parallel to that, we're also starting to become aware over the last few decades just how much genetics contributes to complex trait variation. This is a heroic study from Tinker Poderman and Daniel Postuma, which looked at 50 years of twin studies. And they sort of, over the years of these studies, the picture has become clearer and clearer. Most complex traits have a fairly large genetic component. And so what this does is it invites the question, we know that there's a genetic component to these traits. What are the specific positions in the genome that relate to the trait of interest? Okay. And so that's the principle of gene mapping. We have a priori evidence that there's genetic variation that influences the traits. What are the positions that actually map? How can we dissect the genome to identify the relevant positions? Okay, so the major way in which this began, this endeavor, was through linkage studies. Linkage studies are an extremely good example of using the laws of the Mendelian laws explicitly in their models. So this is a schematic of what you would see if you looked at a pair of siblings. Now, a pair of siblings will share either 0, 1, or 2 alleles, identical by descent from their parents, completely at random due to the random process of meiosis, the recombination during meiosis. Okay, so this random segregation is really useful, because what it's done is it's made a pair of siblings identical in lots of different ways, except for the random allocation of sharing more or less alleles. Now, if any one of these positions related to a trait, when you look at lots and lots of siblings, you'd find that the sibling pairs who share more alleles of that position will be more phenotypically similar to that position. Okay, so it's a direct use of the segregation model from the Mendelian law of segregation. And this is what linkage studies look like. What you do is you get a few markers on a chromosome, sometimes, just a few, sometimes tens, back in the 70s, 80s, and 90s. And you get this low resolution map of positions in the genome that capture these transmitted allele haplotypes that are shared or not shared between siblings. And this was actually an unbelievably successful way to map genetic variants to traits, but not to complex traits. Mendelian traits, which are monogenic, typically, where these where there's a position in the genome that is rare, it segregates in families, and it has a very large effect. Hundreds and thousands of these traits start to become mapped, have their genes mapped very successfully with a good degree of replication. Where it was not successful was with complex traits, multifactorial traits that have many components that influence them, including many genetic factors. The small number that you see here, almost all of those, while published and reported, didn't replicate. Similar to the idea that George presented previously, that candidate gene studies were basically useless. Linkage studies for complex traits, similar situation, extremely poor replication and a crisis in the field. So I just want to bring this back to the laws of the Mendelian laws really quickly, because in order for linkage studies to work and genome wide association studies to work, that I'll talk about next, it's not just that it doesn't use a particular law, it requires that a particular law isn't true. So that's the law of independent assortment. So in order for us to capture the effect of a causal variant, we either have to know what that causal variant is, which is a circular argument. If we knew what it was, we don't need to do the study, or we need to be able to capture its effect using some small number of markers. And the only way in which a small number of markers can capture its effect is if it's correlated with it, if it's co-inherited. And so you observe one marker which is nice and close to the causal variant. So if it's here, this is the causal marker, and this is the causal variant, it's co-inherited. And that violates the law of independent assortment. And it's necessary for linkage analysis to work. So I think that's interesting. It's not a diss on Mendel. You guys have been dissing Mendel a lot, but all right. So linkage studies, abject failure when it comes to complex traits. This is a very influential paper. Risch and Mary Kangas published it in Science in 1996. And basically what they showed was that you adhere to the law of segregation very strongly when you use a linkage model. And that's excellent, but it hampers your statistical power drastically. If you were to relax that assumption and approximate the law of segregation in your model, you could move from linkage to association. And in doing so, you drastically improve your statistical power. What that means is that you can find much smaller effects for the same number of samples, okay? Now, an interesting consequence of moving from linkage studies to association studies is that you introduce a new model. So with a linkage study, you don't really care what the allele frequencies as a causal variant is, because you're looking at transmission within families. If it's only 10 families in the world that have this disease and you have those 10 families, it's fine, because the transmission doesn't care if it's a common or a rare allele. When it comes to association, this becomes exquisitely important, because the power of a detection of an association is related to this term, the genetic variance. And that can be decomposed into two components: the effect size of the variant and the allele frequency, or the variance of the SNP. A rare SNP has a low variance, which means that the genetic variance is low, which means the power is low. So we move into a new sort of paradigm, which is the common disease, common variant hypothesis. So moving from linkage to association requires one big change, which is now we're saying that common diseases which are complex and require sort of very small effects to explain them, those effects, we're going to assume they're common. Because if they're not common, we don't have a chance to find them. There's another really important thing that needs to happen, and that is a major technological advancement in terms of the density of our genotyping. So whereas we're looking at, hundreds of microsatellite markers for linkage studies, when we move to association studies, we're going to depend on hundreds of thousands of variants of genotypes being typed on large samples. Okay, so this is what a genome wide association study looks like. You take samples from the population. You have, in this case, we've got cases and controls. So we've recruited cases for a particular disease. We've recruited controls who don't have that disease to the best of our knowledge. And in this example, we've sort of colored individuals in this sample according to the genotype that they carry at a particular position. And if you squint a little, what you can sort of see is that there's more of the variant homozygote in the cases than in the controls. And so you formalize that difference into a statistical test, which is a drastically more simple statistical test than the linkage analysis. It's just a linear regression. And what you're looking for is a relationship between more variant alleles and higher disease risk. You do this systematically at every position that you can genotype, often 500,000 positions if you're genotyping directly, 10 million positions if you can impute against a sequenced reference panel. And you get a plot that looks like this, and now this is sort of a famous way of depicting the results of a Genome Wide Association study. Really briefly, what this is showing is every point here represents one of, say, 500,000 positions that have been genotyped in the genome across thousands of individuals. And the point on the y-axis, that represents the strength of association of any one of those positions. All right, so you want it to be nice and high. That's a minus log 10 p-value. So nice and high means very many zeros, 0 point many zeros. Canonically, we want it to be less than 10 to the minus 8 in order to surpass multiple testing correction. And this is your gene mapping. These positions that light up with these towers, these are the positions that you are comfortable with declaring to be associated with the trait. And so this started being done in earnest for, from 2005 up until today. Thousands and thousands of these studies have been done. Now, it's really useful to just now go back to the laws of the Mendelian laws and ask what's happening here. So we've moved away from linkage to association. And what's the cost? So this is the cost. So this is a beautiful plot, November et al. 2008 or something, I can't remember, sorry. And what this shows is when you take genetic variants and they drift, when they drift according to demography. Okay, so you can actually use genetic variants to predict the geographic location that an individual comes from. At the same time, different geographic locations have different prevalences of disease. Okay, so when we're looking at the relationship between more A alleles and higher disease risk, we now have a conundrum that we need to solve. Is that because the A allele causes higher risk of disease? Or is that because the A allele coincides with the geographic location where that disease is more prevalent? This is confounding of genetic associations. By moving away from linkage, we're no longer directly modeling segregation. We're approximating it. And when you approximate it, there's a possibility that we have confounding. All right, so the way that this is addressed is you essentially try to use your genetic data to create a proxy for your geographical origin. And so you perform principal component analysis or something similar, which will cluster individuals based on their geographic origin. And you simply fit those as covariates as a proxy for geographic origin. So we're now going to try to factor out any of this bias by hoping that we're capturing that confounding through these fixed effects. Okay, so here's what's happened since the sort of advent of GWAS, so 2005. And look at the scales here. So this is rare monogenic diseases, often like detected using linkage studies. The scale goes up to 4,000. This is common complex diseases. The scale goes up to 80,000. So the number of genetic variants being detected, being replicated, and being considered robust associations for complex disease is truly phenomenal. Like whether or not this is useful, that's a separate question. But the act of mapping genes in this strategy has been an extremely good success from a scientific perspective. I did my PhD at the University of Edinburgh, and we had a very active quantitative genetics journal club. Around 2010, these papers were coming out so regularly, and even at that point, the methods were so well established and so like uncontroversial that these papers were banned from our journal club because they were too boring. And that's in a way like a compliment to those papers. They just, yeah, nothing to see here, just another 20 genes for this complex trait. There is nothing to discuss. And it's quite remarkable, I think. Now, there was another law which I've not touched on yet, which is the law of dominance uniformity. This is an interesting one because a lot of the Mendelian traits do show some degree of dominance. But as we've seen, the vast majority of genetic associations being discovered are not for Mendelian traits, they're for complex traits. And not only do they not show dominance, people don't even bother looking for it. And there's lots of reasons for that. One reason is that to look for dominance, you need 2 degrees of freedom in your test. And when you're so paranoid about losing statistical power, you don't waste a degree of freedom in your test. You just go additive. I think this is a really nice paper because what it does is it sort of quite in a very opinionated style, sort of explains why you shouldn't really worry about non-additive effects like dominance or epistasis. And essentially the idea is if you have a Taylor series of the genetic variation, almost all of the variance goes in the first term. And so your ability to detect the dominance deviation is actually very, very low. And that gets even lower when you don't have the causal variant because the decay in LD gets faster, decays much faster. So this isn't saying that it doesn't exist for these complex small effects. It's just that our ability to detect is very small. And lots of papers have come out that sort of try to point this out. This is a very recent one, and they estimate that you need 20 to 30 times the sample size to be able to detect dominance effects at the same magnitude of simple linear additive effects. So, you know, non-additive effects can certainly exist, but our ability to find them is limited. So this is, again, a law which is kind of ignored quite often. All right. I don't know how long I've been speaking for. I'm just going to keep talking to somebody who drags me off the stage. So there's another crisis comes along every few years. And here's the new crisis, I think this is 2008. So Genome Wide Association studies have been going along nicely. They've been finding variants, they've been replicating. And now we have a new crisis. This is called the crisis of the missing heritability. Okay, so this I don't think that this is melodramatic. I think this is probably almost photographic of how people felt at the time. So here's a diagram of like what the missing heritability represents. So this is the variance of a trait. So this is a complete variance. This is how much of the trait variance we expect to be due to additive genetic variants, right? That's what heritability tells us. And what we're detecting in terms of GWAS hits, those that are significant and that replicate, is a tiny fraction of what we were promised by heritability studies. And so what is going on? Why are we not recapitulating the heritability? And this is important for 2 reasons. One is that a high heritability promises a high possibility to predict the trait. If you can predict 50% of the trait, that's potentially clinically useful. But if you can only predict less than 1% of the trait, that's not going to be useful for anybody for prediction. And the other is that if there's loads and loads of variants, that tells you loads and loads about the potential etiology of the trait. Okay, and you're only finding a small fraction of them. Okay, so I should have put some slides up to illustrate this, but gradually, like many different theories came out about what is the cause of the missing heritability. The major cause is that when you have a highly polygenic trait, meaning that there are many, many causal variants, we're talking tens of thousands of causal variants that each have a tiny effect, then every one of those causal variants is very difficult to detect. You need huge sample sizes to be able to detect them, and you're lucky if you detect any with a small sample size. And so this relates to the infinitesimal model. So Fisher in 1918 put forward this idea of the infinitesimal model, which unified the biometricians and the Mendelian view, that you basically just have a huge number of genetic variants that all act in a Mendelian way in terms of their inheritance, but they have tiny effects and low penetrance. Okay, so such is the promise of GWAS that across the world, governments are now funding these studies. Okay, so in the UK, we have the UK Biobank. That's soon to be cut. That's 500,000 samples that have been linked to electronic health records. That's soon to become Our Future Health. That'll be 5 million samples in the next few years. 23andMe, that's at 12 million samples at the moment. And around the world, there are just biobanks popping up all the time with this hope that you can boost your sample size to be able to detect the genetic variants that underlie complex traits. Okay, so that's the power of GWAS that it's informed these multi-100 million pound investments. So where's that left us in terms of the missing heritability? So this paper is a preprint from Loic Yengo and Peter Vischer and Joel Hirschhorn and others who've been thinking about this for a long time. And what they did was they've been gradually increasing the sample sizes of different traits for GWAS over the years. The last GWAS for height was, I think, 700,000 individuals. This one that just came out, and it's not been published properly yet, 5.4 million samples. And so when you have 5.4 million samples, you have a lot of power to detect extremely tiny effects. And they detect 11,000 independent effects or something like that. And together, those 11,000 effects explain about 40% of the variance. So this sort of idea that, oh gosh, am I being slow? Okay, this idea is important, that you basically need to increase sample sizes to be able to detect these small effects. And if you do, then you will get that. Really important here, almost everything that's been done in the GWAS space has been done in Europeans. And that means that the findings don't necessarily translate to non-Europeans. And I don't think anybody who's a statistical geneticist or an epidemiologist or anything went into that research field with the view that they would only serve one population. So this is something that is becoming realized in the field and becoming addressed. But Melinda Mills and others have sort of started to actually track this. It's quite astonishing just how skewed the data is in terms of lack of diversity and lack of representation across the world. I won't keep talking. I'll just stop. Thank you.